Today we're going to start Unit 3, and there are a series of nervous system lectures, 4 in total, where we'll talk about the nervous system. We'll cover the brain, the spinal cord, we'll talk about the peripheral nerves, and the 4th lecture will work on special senses, and we will mostly focus on vision in that lecture with a little bit of hearing. And then to complete Unit 3, we will talk about the cardiovascular system. So we will have a lecture on the heart and the circulatory system. So that is what Unit 3 is going to entail. So I'm going to start on our first lecture, which is an introduction to the nervous system. And again, there's probably going to be about 3 lectures where we cover the nervous system. There's a lot to go over. So let's do a little introduction first, and then we'll move on. So the nervous system functions. So the main function of the nervous system is communication. So there are electrical signals that are going to connect tissues to the central nervous system. So the central nervous system, which I'll show you on the next slide, is composed of the brain and the spinal cord, and we need to sense everything that's going on in the environment and transmit that information back to our brain so that we can interpret that information. And ultimately, this is going to control and adjust the activity of our body, how we respond to the outside world. And through neurons, which I'll show you the anatomy of the neuron, and we'll talk a little bit about some of the different types of neurons, this will provide a swift but brief response. So we're going to continue to monitor the outside environment. And this is going to work closely with the endocrine system. So this is a system that is going to produce hormones, and this will help regulate all of these functions. All right, so this is an overview of the nervous system, and it's essentially how I'm going to break down the lectures as well. So we'll do an introduction today, and we will begin to start first. We're going to start with the central nervous system. So again, this is the brain and the spinal cord. And then we're going to work toward the peripheral nervous system, which includes, there are cranial nerves, which we'll go over. There are spinal nerves, and then peripheral nerves. And then we'll end by talking about special senses and sensory organs. So this includes hearing, taste, touch, vision, and so forth. But as I mentioned in the first slide, we're going to spend most of our time talking about vision and a little bit of time talking about hearing. We won't really be able to get into some of the other senses, but we'll spend a lot of time on those. All right, so let me show you first an overview. This slide is really nice. It gives you a nice overview of how we're going to work through the nervous system. It's also a slide you're going to probably have to come back to as we work through the 3 lectures. So some of this may make sense to you now, but I'm just going to give a brief overview on some of the topics that we're talking about. And then the idea would be to keep coming back and seeing how all of the pieces fit together. All right, so let me orient you to this slide first. So here we have the central nervous system, which again is, the brain and the spinal cord. This is the place where we are going to integrate, process, and coordinate all the information that we're getting from the outside world. All right, and down here are the receptors that are going to monitor the outside world and communicate this information. So this is the peripheral nervous system. So the peripheral nervous system is anything outside of the central nervous system. So all of these receptors are going to send information to our brain so that we can process that. And that division is called the afferent division, okay? And I like to remember the difference between afferent, and I'll soon talk about efferent, because afferent starts with A, so this information is arriving, it's going toward the brain. So afferent division, all the information from the outside world is going to be sent to our central nervous system for processing. Okay, so that's what I mean by afferent and arriving. And we will begin to work through these different types of receptors. So I already mentioned the special sensory receptors. So we will touch a little bit on this. This is going to monitor smell, taste, vision, balance, and hearing. Again, all of that information will be sent to our brain for interpretation. There are also receptors called visceral sensory receptors. So visceral, if you remember when we were talking about serous membranes, the visceral membrane surrounded the organ. So you can think of visceral as organs. And these receptors are going to monitor our internal organs. So everything that's going on in our body also has to be monitored. So we may not know that information, but that information is still being sent to the brain, such as our heart rate, respiration, all the things that we're not really consciously thinking about. Our brain is still monitoring all of those physiological functions. And then there is somatic sensory receptors, and this is going to monitor skeletal muscles, our joints, the surface of our skin. So one thing to take note of before we get into this, because you're going to see these terms a lot, is this idea of somatic versus visceral. Somatic is anything that we have the ability to have information on, that we know about. Whereas visceral, we do not. So we are not monitoring our internal organs, like I was saying at the beginning, but our brain is. So there's distinctions between somatic sensory and visceral sensory that you should make note of. All right, so we then take all of this information, we send it to the brain for processing, and then something needs to happen. Okay, and again, we're going to talk about all of these different puzzle pieces, but this is the general overview. All right, so then our brain is going to make decisions. And what is going to happen is this information is going to leave the brain through the efferent division. So I like to think of that as exiting. So efferent exiting the brain. And then you get two different divisions here. And one of them is somatic. So this is things, again, that we know about and can sense ourselves. So the somatic nervous system, so let's say we have interpreted something hot is on our hand. We need to move our hand out of the way. So our brain is going to alert our skeletal muscle that is in our arm or forearm and heading towards our hand, and move that and make a movement, okay? There's also going to be a division called the autonomic nervous system. We do not have control over the autonomic nervous system. This is happening in the background, and we will talk about the parasympathetic division and the sympathetic division. Just to give a brief one-sentence overview, this is your fight or flight response or your rest and digest response. And all of our organs, our heart, smooth muscle, all of these are innervated by nerves that are controlling this. So we are always somewhere in the middle. Well, not always. Sometimes we are in fight or flight, and sometimes we are in rest and digest. But a lot of times we're somewhere in the middle. And again, we don't have control over these divisions. This is our internal organs being monitored by the brain at all times. Okay, so that's how we're going to start working with the central nervous system. And then by maybe lecture 3, we're going to get over on this side of the table. And again, if this doesn't make 100% sense to you yet, it will. It's just one of these tables that we show at the beginning, and then you just have to kind of keep coming back to it as I keep filling in the blanks and you learn more about the nervous system. But it's good to see it up front. All right, so let's talk about the organization of nervous tissue, okay? So nervous tissue or neural tissue consists of 2 cell types that we're going to spend some time talking about in this lecture. Neurons is one of them, and neuroglia is the other cell type. So neurons, and they're sometimes called nerve cells, although I don't really like that term, so let us use neurons. They are responsible for the transfer and processing of all of this information in the nervous system that we were talking about. So the peripheral nervous system and the central nervous system have to communicate back and forth with each other. And they're going to do so through neurons. So sensory information is gathered and in the periphery, and it moves to the central nervous system to inform the brain on what is going on. So that is the role of the neuron, and we'll talk about the anatomy of the neuron next. Then we'll move on to neuroglia. Neuroglia are the supporting cells. They are going to be around the neurons to help them with nutrients, to protect them, to help them with their function. That is the role of neuroglia. There are several types of neuroglial cells that we'll talk about, and we'll go over some of their functions. But I want to first start by talking about neurons and the anatomy of neurons. So let's look at a histology slide of neurons first. Oh, wait, not yet. Sorry, I have a little bit more to say about the neuron, then we'll look at a histology slide. So the neuron, as I was saying, this is the basic functional unit of the nervous system. This is how the nervous system is going to function through the neuron. A lot of this I've already said, but I just really want to highlight some of these points. So communication within the nervous system relies on neurons. So this is what the cell type that is going to transmit the information. This is going to happen by electrical and chemical signaling. We won't go through the exact processes by which that occurs because you will get a lot of that in physio, but we'll talk about the basic structure. So we'll talk about the neuron. It has a cell body to it, okay? It contains a nucleus and other organelles just like any other cell, and it has long extensions that convey signals, and I'll show you the structure in a moment. Neurons have a very long life, okay? So the longevity of a neuron, they can live and function for a lifetime. And what's interesting is that for decades, and I mean many, many decades, it was thought that neurons do not divide, and that if you damage neurons, then that's it for you. There's no way to replenish them or regenerate them. All right, so dogma, if you know the word dogma, for what was accepted scientifically, was that whatever neuron, however many neurons you have, that's how many you have for life. So I used to always say, and I even had this on an exam, that neurons do not divide. And then recent studies have shown that there are a few places in the brain where neurons have the ability to regenerate. So I'm telling you this just because of the power of science, and there are things that we can accept for hundreds of years and then find out later by elaborate studies that they may actually not be true. All right, so let's move on here. This is a great histological image. This is a slide of neurons. This is a slice through the brain. So these large cell bodies right here, this is a neuron. They have large cell bodies with processes that stick out and branch in several directions. So you can see all of these branches here coming off of the cell body. These are called neural processes, and they're thread-like material. You can see lots of them everywhere. They have lots of extensions. And then the purple nuclei are the glial cells. So all of these, let me change my pen. I don't know what would work better here. Let's try green. Maybe a little bit better. You can see them, right? All these purple nuclei here, those are the glial cells. And it has been known for some time, although it might recently be disputed, that glial cells, but we're going to stick to it for right now, glial cells outnumber neurons by 10 to 1. So there are 10 neuroglial cells or glial cells to every 1 neuron. Okay, so there are a lot of supporting cells. So you can see, like in this slide, I can count 3 neurons, right? Here's 1, here's 2, here's 3, and then the rest of them are all neuroglial cells. All right, so let's look at the actual anatomy in a cartoon of what is happening with these neurons. All right, this is a nice picture from your textbook showing you the basic anatomy of the neuron. The cell body, right here, is also called, sometimes you'll see it in the textbook or other places, called the soma. So the cell body is called the soma. You will see all the same organelles that you normally see, but just as in muscle cells, there were some specific terms that were only for muscle cells. There is one here too, these Nissl bodies. Nissl bodies are clusters of rough endoplasmic reticulum. There's lots and lots of ribosomes within neurons. This is actually what gives gray matter its color. So you will see there's lots of proteins being made in the neuron, which is why there are so many, why there is a lot of the rough endoplasmic reticulum. All right, you can see, so we just talked about the cell body. You can see the dendrites here. These are long extensions that come off of the cell body. There is also another extension here called the axon. There is definitely a difference between dendrites and axons, and it has to do with the direction of the electrical signal. So neurons communicate, again, by electrical impulses. Dendrites are gathering information and bringing it to the cell body. So this is what is all of these are in the periphery, and they are gathering information and then bringing the information toward the cell body. All right, and then the cell is going to decide what kind of chemical signal to send down the axon. Okay, so here is the axon. This is where the impulse is going to happen. So the cell is going to decide what kind of impulse to send down the axon based on the information that is gathered by the dendrites. And at the end of the axon, so at the very end here, the axon branches. And each one of these is a terminal ending or synaptic ending. It has many other names that you may see in the literature, such as synaptic boutons, synaptic termini. All of these are essentially the same term, terminal endings or synaptic endings. And this is where the neuron is going to communicate with a corresponding cell. And this is going to form a synapse. This is the whole reason why this region right here, this is going to form a synapse. I'm going to show you a closer look at it on the next slide so you can see. But this is where one neuron is communicating with another cell. It could be a variety of things. We'll talk about that in a minute. So lots of times, if this were two neurons, this one would be called this presynaptic neuron, and this one would be the postsynaptic, because it is receiving the signal. All right, let's take a closer look at this synapse. Okay, so again, the terminal endings, this whole region right here is going to create a synapse. Okay, so let's take a closer look at what this synapse is. So if you take a closer look right here, this small box of where the terminal endings are attaching to the next cell, and I'll explain what that next cell might be. It could be a neuron. There is what is called a synapse. And at this synapse, there are chemicals that are being released to the next cell. So if we are going to call this a synaptic bouton, what is happening is there is a release of chemicals called neurotransmitters, which are then going to signal a receiving cell to do something. So again, this would be the presynaptic cell, and then the next one would be the postsynaptic. And you can see the neurotransmitters are in these vesicles, and then they are released right here onto the next cell, so that that next cell knows what to do. So neurons can synapse with a variety of other cells. So it can synapse with another neuron. So neurons communicate with each other through this synapse process. Neurons can also communicate with skeletal muscle. Okay, so if we need movement to occur, the neuron can communicate with skeletal muscle. It can also communicate with smooth muscle or cardiac muscle. And then finally, neurons can synapse with gland cells. So this is going to tell a gland whether to secrete more of something or whether to secrete less of something. So now let's move on and talk about the different types of neurons. All right. So again, we're going to talk about the anatomy of these neurons, but we're not really going to talk about how they signal. Okay, it's a complicated process that you will learn about in physio, but we're limited here to just stick to the anatomy of these different types of neurons. So there are 4 basic types of neurons. All right, the first one is anaxonic neuron. Okay, anaxonic. So you remember what avascular means, right? Without vascularity. Anaxonic is without an axon. So this is without an axon. Anaxonic. So this type of neuron is actually extremely rare and not well understood. And it actually does have an axon, but it is impossible to determine which is an axon and which is the dendrites, right? You can't determine that just by looking at this. So we're not sure which axon or which of the dendrites, which of any of these cell extensions are sending information to the cell and which ones are sending information out of the cell. It's not obvious here. So that is what an anaxonic neuron is. And again, they're not very well understood. They're only found in the central nervous system, and they're found associated with special senses. All right, so that's that one. The second one is called a bipolar neuron, and this is also very uncommon. This is also uncommon. So the dendrites converge into a single dendrite. So you can see the dendrites converging into a single dendrite that then feeds information to the cell body. And then there's a single axon which will relay the information out. So this is called bipolar because there are two poles and the body or the soma is in the middle. And this is very rare too. It is only found in the brain, so in the central nervous system. And it is also associated with special senses, in particular vision. The most common type of neuron is this third one, which is the pseudo-unipolar neuron. This is similar to the bipolar. So the signal from the dendrites continues on through the axon and doesn't stop at the cell body though. There is a cell body there, but it's off to the side. So you can see it here. So it is found associated with the peripheral nervous system. So most all of our sensory neurons are this type. So this is sensory neurons. And so the dendrites are gathering information. All of this information is fed to the body. And so what normally happens is what I was just saying, that there are dendrites, they gather information, all the information is sent to the body, and then the body makes a decision, and that decision goes down the axon. Well, this is not what is happening here. So because we need to take in all the sensory information that's going on in the outside world, we don't want the cell body making decisions for us. We want to send all of that information to our brain and let our brain figure that out. So this way, with the cell body off to the side, there's no chance for the cell body to determine what it should and what it shouldn't be, what should and shouldn't be sent. That information is just going to travel all the way down. All of the information that's gathered by the dendrites will then be sent down the axon. And this is going to be touch, temperature, pain, all of these types of senses from the outside environment is going to occur through the pseudo-unipolar neuron. All right, and then the fourth one is the multipolar neuron. This is the single most common type of neuron. This is the neuron that I showed you at the very beginning of this lecture. All motor neurons are this type. All right, so this is going to decide whether a muscle cell is going to contract, yes or no. So there's lots of information coming in from the dendrites. It's going to be transmitted down the axon, and then it's going to connect with skeletal muscle. So this is multipolar, meaning there's lots of poles here coming in with a single pole coming out. All right, so we just looked at the structural classification of neurons. Now let's take a look at the functional classification of neurons. So we need to classify neurons by what they do. And different neurons, based on where they are, have different roles. So there's two types of functional classifications, somatic and visceral. And I touched on this when I was pointing out, sorry, I need to silence that. When I was going over the overview chart of how I was going to break down the nervous system, I gave you a little bit of a sense of what somatic and visceral meant, but I wanted to also put a slide in here so that you can see this. So again, somatic tissue is anything that we have voluntary control over. like our skeletal muscles. All right, we are also consciously aware of sensation. All right, so skeletal muscle, and then consciously aware of sensations. So this is pain, touch, and temperature. And this is defined as somatic. And then, as I was referring to before, visceral means that we have no conscious awareness, or we have no control over these our internal organs. So this could be like asking your erector pili muscle to contract to raise a single hair. No matter how hard we try, we can't make that happen. Or telling a blood vessel to constrict, right? We can't make that happen. So visceral, motor, and sensory have to do with things that are not in our control. So motor signals to involuntary muscles, such as smooth muscle and cardiac and glands. Again, remember that this has to do with the autonomic nervous system. All right, we are not consciously aware, but our brain needs to keep track of everything that's going on in our body. The same is true for sensory signals. So this is like, do we know the pH of our stomach? We don't know it, but our brain knows it. So there are all these things that are being monitored, whether it be movement or sensory, that we have no control over. And you have to just get used to using this terminology. You have to get used to understanding what somatic sensory, somatic motor, visceral sensory, and visceral motor means, because we're going to use these terms throughout the nervous system lectures. So it's important to have a good understanding of these terms sooner than later. All right, so let's talk about the types of signals that are being conveyed. All right, so I also touched on this in the very beginning, the introductory slides. So we have sensory neurons that are going to send information from the peripheral, so the periphery, from the outside world, and it's going to take that information to the central nervous system to be processed. So we have somatic sensory, and we also have visceral sensory. Again, somatic we have control over, visceral we do not. And then we have motor neurons that are going to process this information, or after it's being processed, the information is going to get sent from the central nervous system back to the periphery. And again, I gave you that example of there being something hot on our hand, and we would need to move our hand out of the way. So that is somatic motor. Visceral motor, that's autonomic. We don't have control over that, but our brain is still interpreting that information for us. So again, you're going to have sensory neurons that's going to send information to the central nervous system, and that's going to lead a response, and you're going to then have motor neurons to deal with that response. There is something called an interneuron. So interneurons are neither sensory or motor, all right? So what is happening is that we have to make decisions. So let's say, and we have to make these decisions because we have to coordinate the activity. We have to make sense of what's going on. So for instance, here is a job of the interneuron. So let's say you're getting a needle injection. We know that there is pain that's going to be involved there. But at the same time, we don't want to pull away from that because that might cause the clinician to have to stab us twice with the needle, right? So we're going to make a conscious decision not to move away. On the contrary, if we're being stabbed with a knife, we want to pull away. So the brain is making decisions on what to do with the information that's being sent to it. So that's what I'm referring to here, that these interneurons are going to analyze the sensory input and coordinate the proper motor output, all right? Now that was a really good example for somatic sensory and somatic motor, but you might have a little bit of a harder time understanding visceral sensory and visceral motor. So let's say you are out for a run and you need to take in more oxygen through your airway. So your visceral sensory neurons are going to recognize the fact that you need more oxygen and more air running through those airways. That's going to send a signal to the brain, and then the visceral motor is going to change perhaps the diameter of those airways. Maybe it will dilate them so that you can take in more air. Again, we can't tell our lungs to do that, but that is how everything that we're doing is being perceived by the brain, and then adjustments are made according to our activity. So hopefully that's clear. I know it can get a little bit confusing, and we'll keep talking about these terms and these ideas so that you can get used to talking about them as well. Make sure to look at this in your book if you're having a hard time understanding this. And just one other note, this is a complicated pathway, and we're just going over the very basics of this. You are going to get a lot more of a better understanding of the types of signals and how this works in physiology. But we just want to give you a basic understanding of some of these terms before you get there. All right, so now we're going to talk about the cellular organization of neural tissue. And we're going to talk about the support cells that are in both the central nervous system and the peripheral nervous system. So be sure to have diagrams that you're working out either in your notes or in some kind of format in your study guide so that you can keep all of this straight. So we're going to go through the types of support cells, those glial cells that I had mentioned at the beginning. We're going to talk about the different populations of them and what they do. So there are two populations of neuroglia. One is in the central nervous system and one is in the peripheral nervous system. All right. We're going to start by talking about neuroglia in the central nervous system, and then I'll switch over and talk about the peripheral nervous system. So we're going to first talk about the four main types in the central nervous system. Those include astrocytes, oligodendrocytes, microglia, and ependymal cells. All right, and you can see a nice picture here, which would be good to put in your notes so that you can keep track of where these cells are and what their functions are. All right, so let us start. Here is a nice overview of the central nervous system. We are going to start with the oligodendrocytes. So this is an image taken from your book, and we're going to work through each of these and talk about them on their own slides. So let's first start with oligodendrocytes. So oligodendrocytes, they form a scaffolding. Okay, so the nucleus of the oligodendrocyte, let me show it to you. So here, let's look at this picture first. So here is the nucleus of the oligodendrocyte. And you can see that there are processes off of the nucleus that reach out to various axons from different neurons. Okay, so here's an axon here, another one here, another one here. So they're reaching out to different axons, and they're holding them together so that they can't freely move around relative to each other. So do you see these processes here? So they're reaching out and grabbing on to different axons from different neurons. And you can see another picture here of the oligodendrocyte. Here are a bundle of 3 axons right here, and you can see that it is grabbing on to 3 of these. And this forms kind of a scaffolding. Each process ends by wrapping around the axon. So you can see a little bit of the axon still here in yellow, right there, and a little bit over here. But you can see that they're wrapping around, all right? So it is wrapping around in repeated layers of a membrane with a material in it called myelin. So what myelin is doing is it is insulating the axon and the electrical signal, like insulation you would find on wiring in your house. And by wrapping around the axon, it actually speeds up how fast the electrical signal can travel. So this wrapping of myelin is what actually creates the white matter that we see in the brain. So it provides a structural framework, but it also helps with the speed of the signal. So one oligodendrocyte wraps around multiple axons. So it creates a framework, but it also helps the neurons reach their full potential as it can control the speed of the signal. So yes, it provides a framework, but it also helps with the speed of the electrical signal. So it acts as insulation. The next type of glial cell that we're going to look at is called the astrocyte. And the astrocyte gets its name because astro means star, and they look like a star. So you can see this image right here. They have all of these processes coming out in all different directions from the main body. There's lots of them. And if you take a closer look at them up here, they have these foot processes called perivascular feet, right here, or foot processes. So part of the process will attach to the body or the soma of a neuron, so you can see that here, and the other part will attach and cover blood vessels. You can see the capillary here. And they are providing, so their function is to provide a structural framework by holding a neuron and a capillary together. So look at them here. The astrocyte is holding the neuron and the capillary together. Now, those first 2 pictures quite aren't, they're not quite an accurate representation. Up here is a better representation of what is happening. So each of these blue areas right here, each of these, I guess it's kind of hard to see with my color, but you get the idea here. Each of these is a foot process. And you will notice a number of them. So realistically, there are no gaps on these blood vessels. Every blood vessel that penetrates into the central nervous system, the entire surface of that capillary is covered with these foot processes of the astrocyte. And they are ensuring that what is inside this vessel, so you can see here inside this vessel, there's going to be blood, and another fraction of blood is called plasma. And within plasma, there are things like oxygen and glucose, and amino acids. And we want to make sure those things don't escape. So there's also endothelial cells. So there are these cells that line the blood vessel right here. These are called endothelial cells. They are simple squamous cells. They have gap junctions to the next cell. So this cell over here, there's a gap junction to hold these cells together and to ensure that nothing escapes. And that is also what these foot processes are doing. So anything that ends up in the extracellular fluid out here is going to be highly regulated by the astrocyte, okay? And this is what is creating the blood-brain barrier. And sometimes you'll see it referred to as the BBB. And this is what is going to be protecting the nervous tissue. So this is also a major barrier to bacteria, viruses, or other pathogens, okay? We don't want any of that to enter our brain tissue. And so the reason for this is that the blood-brain barrier keeps almost all of that out of the brain. So astrocytes can regulate ions and what gases get through. They can recycle neurotransmitters, and they can also repair if there's damage. So they have a lot of functions, but the main function is to create this blood-brain barrier. The last two types of cells are called microglia and ependymal cells. Okay, so microglia cells are on the left, the ependymal cells are on the right. Let's talk about microglia first. So remember that I said that the blood-brain barrier is to keep pathogens out. However, sometimes they do get past the blood-brain barrier. And if they do, the last line of defense are the microglial cells. They are immune cells, and they can phagocytize, meaning they can gobble things up just like macrophages do. So macrophages are the normal cell type. Macrophage are the normal immune cells that can gobble things up that shouldn't be there. Microglia are similar, but they're much weaker than a macrophage, like comparing the two, but they are there as host defense. So they can remove pathogens if something has escaped the blood-brain barrier. All right, let's talk. So it removes debris, waste, and pathogens by phagocytosis. Did I go over all of these here? Let me just make sure. The astrocytes maintain the blood-brain barrier. There is some structural support. They do regulate ions, nutrients. Okay, we went over all that. Good. I just forgot to underline it, so I wanted to go back and make sure. The ependymal cells. These are a little harder to explain, and you're going to understand this better when we start talking about cerebral spinal fluid, which we're not really going to do in this lecture, but you need to know a little something about these cells so when we get to the cerebral spinal fluid, it'll all come together. So these ependymal cells line fluid-filled spaces in the brain and the spinal cord, which contain cerebral spinal fluid. So cerebral spinal fluid. And these are specialized, or cerebral spinal fluid, so also called CSF, is a derivative of plasma, and it is there to provide nutrients. It's there for waste exchange, and it is within nervous tissue throughout the brain. And we'll talk about exactly where you will find it in a later lecture. But for instance, in the spinal cord, there is a central canal that runs through the center of the spinal cord, and cerebral spinal fluid is located within that canal. And interestingly, on the surface of the ependymal cells are cilia. Let me get a better color here. Let's try green. You can see all the cilia. I'm just going to write it here so that you know. This is cilia. There is cilia on the apical surface of these cells. And the reason for this is to be able to move the cerebral spinal fluid around. That is how it's going to circulate, is from the cilia here on the surface. I kind of drew that too far. So we will talk, there are actually ventricles inside of the brain where this fluid is going to flow. I just mentioned the central canal of the spinal cord, and these cells are going to assist in the production and circulation of the cerebrospinal fluid. All right, this is another great picture from your textbook that gives you a sense of the overall organization that is occurring in the brain. This happens to be the spinal cord, but it would be the same in brain tissue as well. So there are two areas. There's what's called gray matter, and there's what's called white matter. The gray matter has the large cell bodies. You can see them here and here. There's two of them. And these cell bodies, remember, are also called, oh shoot, I spelled it wrong. Are also called the soma. All right, so these are the large cell bodies, the soma of the neurons. They have those Nissl bodies that give the gray matter their gray coloration. And then we have white matter. White matter is composed of the axons and the glial cells that support them. So cells don't have, the support cells, the glial cells, don't have the Nissl bodies. So there's no gray coloration here. So you can see the oligodendrocytes reaching out and holding on to two adjacent axons. So here's the oligodendrocyte. You can see it's holding on to two different axons here. You can see that there is myelination, so it is making myelin right here. You can see an astrocyte holding a blood vessel. Here's the astrocyte. It's holding on to the blood vessel and an axon. So here's the axon. Here's the blood vessel over here. And this is actually an accurate representation of the foot processes and what the surface of the blood vessel is going to look like. It's going to cover the entire surface. And then each one of these gaps in here, right, you can kind of see where I'm drawing, are held together by gap junctions. You can see the microglia. Here's the microglia. These cells are moving around. They're going to be mobile. So they're moving around in the tissue, gobbling up pathogens or cellular debris. And then you can finally here, you can see the ependymal cells. These are lining, let's say, the central canal, which has cerebrospinal fluid in it. And the picture doesn't show the cilia on the surface here. I'm going to kind of draw them on there for you. But this gives a really nice representation of what's going on. It's actually even more compact than what this picture is showing you, but you get the general idea of what is going on. There's lots of cells, there's lots of processes going in many different directions. All right, so now we're going to switch. We just talked about the cell types in the central nervous system. Now we're going to switch and talk about cells in the peripheral nervous system. So let's highlight that. There are two cell types that I need you to know about that are in the peripheral nervous system. One of them is called satellite cells, and one of them is called Schwann cells. So we're going to start with the satellite cells. All right, here is a picture of the satellite cells. And what these do, these cells are completely covering, so here they are, the cell body. All right, so here is the cell body. Whoops, didn't draw that very well, but that's the cell body, okay? And it is going to cover, these cells are going to cover the entire surface. So you can see the axon kind of running along here. Do you see the axon running along here? And it is covered in myelin. And the cell body is off to one side. Do you remember what kind of neuron that is? So that is the pseudo unipolar neuron, and that is a sensory neuron. Remember, we talked about that. If you need to revisit that slide, you can. So the cell body is completely covered to keep it protected from the extracellular fluid. So you can see here's another image of it completely being covered. And so it is going to monitor what has access to that cell membrane of the neuron. Okay, so it's going to surround the neuron, the cell body, and it's going to regulate gases and nutrients and what that cell body has access to. The last cell we're going to talk about is the Schwann cell. All right. The Schwann cell is responsible for myelination of the axons. Now, not all axons are myelinated. Most of them are. And it's doing it a little bit differently than the oligodendrocyte that I talked about earlier. All right. So what's happening here is that, let me show you, is that there's no connection to multiple axons like before. Instead, each one of these segments is a result. So each one of these segments here, 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 and here, each one of those segments is a result of a single Schwann cell that wraps its cytoplasm around and around, creating layers. So you can see that is what's happening here. The nucleus of the Schwann cell is right here, okay? And if you follow its membrane all the way around, you can see it wrapping around the axon over and over. And that is what is creating myelin sheath. So here is one, and this is another one over here. So in this instance, each Schwann cell only covers one small region of one axon, and it doesn't connect adjacent axons. So be sure to be clear on the difference between the oligodendrocytes and the Schwann cells. The purpose is still the same. You're still going to get protection, and you're still going to get insulation that's going to allow the electrical impulse to speed up. So Schwann cells begin to wrap around, so you can kind of see, let me show you what's happening here. So here you can see the Schwann cell begin to wrap around, so the edge keeps going. So here you can see this edge right here is going to keep wrapping around and around, and it's going to push all the way around. The area and the spaces are going to fill with myelin. So that's what you're seeing here. You're getting this area that is going to fill up with myelin. And the result here is a fully myelinated axon. And the nucleus is usually pushed off to the side here. We have a really nice model in the lab of a neuron that is myelinated, so you can see the anatomy and look at it. We'll use that model in the lab to study from. So that's the process of how a Schwann cell myelinates the axon. And the last topic that we're going to cover in this lecture is the anatomy of a peripheral nerve. Okay, so this is what this is depicting right here. So the nerve is considered an organ, and I never really defined this before, and I'm not going to test you on it, but because I meant to mention this earlier on. An organ is something that consists of all four tissue types that we talked about probably day 1 or day 2. So it contains nervous tissue, it contains epithelial tissue because, as I mentioned, there were endothelial cells that were on blood vessels in the last slide, so there is epithelial tissue. It also contains smooth muscle tissue because of the capillaries. There's smooth muscle around the capillary, and then there's blood vessels around the blood vessels, and then you have connective tissue. So you have all of those tissues. When you have all of those tissues present, that's what makes an organ. Okay, anyway, so that's a little bit of FYI. It's too late for me to worry about that, but I did want to pass that information around. So the terminology, the way that a nerve is organized is a lot like what we saw with muscle. Okay, you're going to see a lot of the same terminology here. So if you remember how a muscle is organized, this should come relatively easy. So this entire bundle is a nerve. One nerve. One nerve has many individual axons in it, both sensory and motor. There are no cell bodies here, okay? There are no cell bodies outside of the spinal cord or the brain. So these are just the axons. So around the outside of a nerve is connective tissue called the epineurium. So remember, it was the epimysium in muscle. So mysium was muscle. Neurium is for a nerve. So if you can kind of remember epimysium and epineurium. Okay, so what is surrounding the nerve is epineurium. And then axons are bundled together like fascicles. Remember the term fascicle for muscle? The same thing is happening in nerves. And there is connective tissue that is surrounding the fascicle. All right, and this is called the perineurium. Just like the perimysium, you have the perineurium. And inside the fascicle, you have lots of individual axons. Okay, so if we pull that out, you can begin to see some of these axons that are in there. These individual axons also have Schwann cells associated with them. And then there is connective tissue shown right here that is surrounding both the axon and the Schwann cell, and that is called the endoneurium. Okay, so we're changing mesium for neurium. And my last slide is a really cool scanning electron microscope image. It doesn't show the epineurium because we're not on the outside. This is a nerve, but we're more on the inside of the nerve. But you can see, let me change my, you can see here is the perineurium, and then the endoneurium is what is going to surround each of these individual axons. And axons come in different sizes, and they have different amounts of myelin on them. You will work that out when you take physiology. You can also see some blood vessels here, some really large blood vessels here. You'll see them inside and outside of the fascicle. So here's a couple other ones. Some of them are inside, some of them are outside. All right. That concludes the lecture on neural tissue. We're going to move into the spinal cord and get into some more details on brain anatomy as well. I will talk to you and see you in the next lecture.